Hello, hello, Barshan and Morgan. How are you? Did you make a pull request, or is that just the blog post? All right. Did you want to share that with us, even though the?
So, so my main goal was like this is what my outline looked like before this week. Like take a process bio, pass it through the model, computer loss, backpropagate, and save a checkpoint. And so that it goes on and we have a good accuracy. So one important part was training the wrapper around the dataset. Like it only contained embryo, so I also added a small label wrapper that attaches the correct class when each sample is loaded. So since we are predicting the classes at the end of the whole pipeline, so I attach the label to the npz file so that we have that testing scenario going on. So then we have a train and validation split, so the classes stay balanced, and the class weighted cross entropy to handle the imbalance automatically. Like these are basically like I had like a 20 to 80% of the classes, and so most training settings are configured through command line arguments, and which makes it easier to run and run different experiments without changing the code. So so that I can run each like the change the parameters and whatever the window size from the command line itself. So the training loop was straightforward. It's like it loaded an embryo, ran a forward pass, and computed loss and backpropagated. Then it clips the gradients and updates the weights. So I also added like checkpointing so that if I run it for a whole day, so it just checkpoints. Uh, or if the system crashes, we still have a checkpoint to go to, and the weights are saved. So the first mock test was small, just 4 embryos for one epoch, but it's enough to confirm that the whole pipeline works. The accuracy was around 50%, which expected for a random initialization on such a tiny run. But the important part is that the all everything was running properly. So as you can see, like on the first epoch, there was a train and loss of 0.69 and the accuracy of 0.25 on the train data set, and the validation it was zero point 0.5. And it ran for like 13 seconds, one epoch. So, yeah.
That's pretty good. Yeah, thank you. Did you have more, or I
No,
thought there was
like,
more down there.
no, 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 no. That is
Okay.
it.
Well, that's good. Thank you. Yeah. Nice update. So yeah, I don't know. We get started here. I have something I wanted to go over that I was working on. This is kind of a follow up to some of the things that we were working on for uh, our actually our cancer physics book, and that's sort of in press right now. So uh, I want to go over kind of what I've been working on. It's very kind of early days on this, but um, we'll take a look. Then I wanted to go over some papers. Wanted to follow up on the last paper I presented last week, and then a few more things. So this is a notebook I'm building with the assistance of Gemini, and this is called Perlin Noise as a model of biological tissues. And so the backstory of this is that a couple years ago, um, Richard Gordon asked me to do some Perlin noise demonstrations during DevoWorm, and the idea was we were going to create Perlin noise, or we were going to like build some code for Perlin noise to generate a background for masking tumors in biological images. So, if you have like a biomedical image of a tumor, oftentimes it's embedded in noise and you have to find ways to extract it from that noise. And so the whole embryo physics book is about using different types of techniques, different types of imaging techniques, X-ray techniques to get at that image or to get at the tumor despite noise. And so the Perlin noise is supposed to be the thing that sort of masked that signal. And so eventually the chapter got written. It's a chapter called Perlin noise and how to avoid it. And the point of that chapter in context is that Perlin noise is a good way to model biological tissue. So if you're not familiar with Perlin noise is a procedural type of noise. It's procedurally generated, and it was first developed in the computer graphics community. And it's something you often see in computer graphics, video games, etc., where you see different types of noise that are generated. And so, the idea is that you have this algorithm that generates noise. It's not random, it's not Gaussian noise, it's random noise, but it has some structure in it. And so the idea of that chapter was that Perlin noise is good at modeling different types of biological tissues, or maybe a very generic biological tissue. So if you have a tumor embedded in some biological tissue and you want to be able to detect it using an X-ray methodology or something like that, you should be able to pick it out using an algorithm that picks it out of that noise. And so what this notebook is then is the follow-up to that. So the follow-up is that if we assume that Perlin noise is a good model of generic biological tissue, then we can expand that to different types of tissue and model Perlin noise as this 3D volume, and then maybe use some techniques to optimize that. So one of the reasons Perlin noise is good at modeling tissue is because it has this sort of non-Gaussian distribution. So you have Perlin noise. I'll show you in the first cell what an example of Perlin noise is. But basically it has sort of a quasi structure to it. And so that's kind of the idea of why it looks like tissue. If we go back to when we've been talking about the organization of tissues, we know that cells migrate around the tissue, but they form these layers, and the layers are, sometimes non-uniform. And so that's what we're getting at with this modeling exercise. So in consultation with Gemini, I kind of came up with a methodology for what I wanted to do. So the first thing I want to do is generate a 500 by 500 array of Perlin noise. And we'll see what Perlin noise looks like. So it's a two-dimensional array. So Perlin noise is generated usually in 2 dimensions. If you use the standard Perlin noise techniques, say in Python, it's a 2-dimensional array. And you can't really build a 1-dimensional array of Perlin noise because, as it turns out, the noise is generated sort of row by, or it's column by column down, and it's the depth of the second dimension is the generative process. So that's actually time. So you could take like a single slice of Perlin noise and say it represents any one time period, but that's not really the way to look at Perlin noise. You have to look at it as a 2-dimensional array. So we need to have a 2-dimensional array of Perlin noise just to see what it looks like. And then we might want to expand that to an n-dimensional case where we have multiple dimensions of Perlin noise. Then we have visualizing this in like an anatomical slicer or some sort of simulated way of viewing this, much as an anatomist would do. So having this volume of Perlin noise and then cutting it at different orientations, and then looking at that surface cut at that orientation, and then you can view different aspects of the noise. In this case, an analog of some sort of tissue. Step number 4 is to implement a genetic algorithm to optimize for multiple tissue types or textures. So this is where you're going to have different types of tissues simulated, and the genetic algorithm really just optimizes for those different tissue types. Then we use something called the GLCM, implement that and visualize the textures. And then I did this replacing the GLCM with a spatial pooler. So the GLCM, as we'll see, is a computer graphics tool, and we'll get to the notes on that in a little bit. But basically, we want to visualize the textures using an alternate method, and then we want to use a spatial pooler, which is a spatial algorithm or a spatial encoder to represent that. And then these step 7 and 8, which I couldn't get fully to run yet. The first, number 7, is generate a volume of 3D Perlin noise using a GAN, or a generative adversarial network. So instead of the genetic algorithm, I want to use a GAN as an alternate form. And then I want to visualize GAN-generated Perlin volumes via anatomical plane. So I basically want to replicate what I did up here, but with a GAN. And GANs are, they were really hot about 7 or 8 years ago, but they're not really used much anymore. But they really offer this nice compromise between a generative model and a discriminative model. And anyways, I haven't been able to get it to run yet, so I can't show you the payoff there, but we'll go through the notebook step by step. So the first step is we want to generate a 500 by 500 pixel array of Perlin noise. So we have a Perlin noise function in NumPy, I believe. This is where we have this function Perlin noise, and you could define the parameters here. You have these grid coordinates, random gradient vectors, and then let's see, and then we generate our noise. So here's our noise, and this is what Perlin noise looks like, and it's actually tunable. So you can tune a field of Perlin noise in different ways. This is very blurry. I can play with some of these parameters up here. I'm not going to do that now because I don't want to waste too much time on this. But basically, you can change the parameters. You can tighten up the field of noise. You can make it blurry. You can, make it sort of more sharp. You can do all sorts of things to the noise. And of course, this is what we're describing in the chapter as this analog to biological tissue. So you can see that it's sort of this non-uniform type of noise. It has these structures within it. They might look like things like areas of cells, even kind of, it's really, you could describe tissue in this way. Let's just put it that way. Okay, so that's the two-dimensional volume. And you'd start at the first, I guess you start at the first row or column, and then you just continue across the two-dimensional volume as the generative process unfolds. And so that's what you have with your two-dimensional volume. Then you want to expand this to n dimensions. So expanding this to n dimensions, we want to have this this third dimension or fourth dimension. We're creating a mesh grid, and we're creating random gradient vectors at the lattice points. We're computing dot products for all 2 to the power of dimensions corners. We're generating all corner combinations, and then we're normalizing this. Then we have this variable noise 2D, which is where we have, this is the 2-dimensional case, this is the 3-dimensional case. So now we're going to visualize a slice of the 3-dimensional Perlin noise. So this is where we have three-dimensional noise slice. And I'm not sure what it was doing. I guess it was taking a slice through the 3-dimensional surface. And it kind of looks like this. I'm not sure this is good, kind of what I want. But it does give you that 3-dimensional, what it would look like in 3 dimensions. So this, and I think this is like a z of 20. So the z dimension has a value of 20. And you can change this accordingly. So what parameter is that up here? Somewhere up here. Anyways, I'm not going to waste time on that. Anyways, you can tune that as well. So you can get different numbers. I haven't tuned any of this really yet, but I'm going to play with it a little bit more as we go along. So that's what the 3D case looks like. And my impression of this is that it's a bunch of different generative processes within this 2-dimensional field. Okay, so then we have the different slices. So actually we're slicing through different parts of the Z volume. So this is Z equals 10, Z equals 20, Z equals 30. And it just shows that you're kind of going through, and you saw in the 2-dimensional case, where you have this non-uniform noise, and you see the non-uniform noise carry over a little bit into this Z slice, where it kind of looks the same in terms of the patterning, but it isn't. It's slightly variable. So that's kind of the idea. We'll play with it a little bit more and see what it looks like. So then we want to build, remember, we want to go from the multiple n dimensions, in this case 3 dimensions, to some sort of genetic algorithm. So this is where we build an artificial genome. We have these parameters: scale, octaves. These are things that are specific to tuning a field of Perlin noise: persistence, lacunarity, anisotropy, and angle. So all those things are involved in the genetic algorithm. And those are the things that are going to be optimized within the genetic algorithm. Tissue types, we're going to aim for fat, muscle, trabecular bone, and these are going to have different target textures. So each of these parameters are going to have a certain value that's optimized for these different tissue types. The fitness is going to be where we match the target mean, standard deviation, anisotropy, and frequency of gradients. And then the output is the best texture per tissue plus visualization. So for every tissue type, we're going to have a best texture. Okay, so this is where we get our two-dimensional Perlin noise field with simple anisotropy and rotation. We're going to define that here. Our anisotropy is 1.0. Our angle is 0.0. So then we go through this. We have our base grid. We apply anisotropy and rotation in coordinate space. We have our integer lattice. Then we determine min-max values for integers to ensure non-negative indexing, just because we're iterating through this and we don't want to end up with something that isn't within the bounds. Then we have our random gradients. Then we determine the maximum index required for gradients based on shifted coordinates. This is where we're getting into our genetic algorithm. Let's see. We interpolate the weights, normalize to 0 to 1. And then we have this function FBM Perlin 2D, which has all of these different parameters for the genetic algorithm. This is fractal Brownian motion over Perlin noise, so we're introducing this sort of evolvable aspect to the Perlin noise. And we have an amplitude of 1, the frequency of 1, total amplitude of 0. Let's see. Then we have our texture features. So then we're going to define some texture features and our tissue profiles. So this is where we have the fat, the muscle, and the trabecular bone. So again, our parameters are different for the different types of tissue. So fat is basically smooth, isotropic, and low frequency. You'll see this in the visualizations. The muscle is anisotropic and striated. So isotropic just means that there isn't a lot of variation over space. Anisotropic means there's variation over space. In muscles, you have these striated bands, as opposed to fat, where it's just kind of this almost like a gelatin. And then there's some variation, but it's not very common. And then trabecular bone, where there's more contrast and there's a moderate amount of anisotropy. So that's what we're aiming for here. Then our genetic algorithm is here, where we have our parametric bounds for these different parameters. We have our function for random individuals, so we have scale, octaves, persistence, lacunarity, anisotropy, and angle. We then have our mutation function, which is at a rate of 0.2. And so that means that we do our genetic algorithm. I have to say that, like, when Gemini generates a genetic algorithm, it's kind of interesting how it does this. It doesn't give a lot of detail as to the sort of this specific methodologies for it, but it does produce this sort of very simple genetic algorithm. So it has this mutation rate, and it just determines, what gets replicated and what the mutation rate is here. Then we have our crossover operator here in this function. Then we have individual to noise, which is this function here. We have our fitness function, which is here. And this is L2 distance and feature space. So it's using this as sort of the norm for determining fitness. And then we have run GA for tissue. I guess we're running that. And then we're running each one. We're running a GA for each kind of tissue, fat, muscle, and trabecular bone. So that's what we're doing. Then our visualization, we want to visualize our results. So now we're getting our best texture plus our fitness curves. And so that is here. So we run for, I guess, 39 generations, which isn't a lot, but it gives us this best fitness of 0.1705. So the algorithm converges here at 39. And so you can see the graph here where for each tissue type, you have a fitness function that converges at around 10 generations. For trabecular bone, it's a little longer. For fat, we actually expect it to be really short because it's not evolving very much. And you can see in fat, you have this limited anisotropy. You have kind of like these bands. And then you have these gray areas, which is like what fat might look like. Muscle is this here. I mean, it's very blurry, but you can see sort of these striations. A little bit takes a little bit longer to converge, and then the trabecular bone, where it's a little bit more frequent than fat, but it's it takes about 20 generations to converge to this. So that's kind of what we've been able to evolve on this. I think that needs more work to get that right. And remember when we originally thought that this was an analog for tissue types, we're talking about, kind of like it provides sort of the statistical structure of a tissue, not necessarily that it's going to look exactly like tissues you identify in different biological contexts. I have some better visualizations of evolved tissues. I don't have them in this presentation, but I can get them out. I'll be presenting on this later. Let's just put it that way. Okay, so that is about our genetic algorithms. I like that. I think it's an interesting set of experiments. It needs a little bit more work on maybe the starting point of the Perlin noise. But anyways, that's what that is. Now we move on to this GLCM, or the gray level co-occurrence matrix. So this is something that's used in computer graphics as this sort of texture method that quantifies texture analysis of pairs of pixel intensity values with specific spatial relationships. So basically, it takes these textures and it gives us some sort of statistics on them. GLCM allows for the genetic algorithm to evolve Perlin-based textures and replicate biologically realistic microstructures. GLCM captures local spatial dependence, directional structure, which is critical for muscle, collagen, and trabecula, contrast and homogeneity, entropy and complexity. And then this is what it's doing here. It's putting in this GLCM metric to the genetic algorithm fitness function. So this is where we're using skimage to import these tools for GLCM. We have this texture features GLCM function, which is how we're going to compute all these statistics. We're going to convert things to 8-bit environment. And then we run that, return a vector. Then we have our tissue profiles again. We're refining those for fat, muscle, and trabecular bone. And then we have this fitness function here, which is defined as this L2 norm again. So then, GLCM captures second order spatial statistics, which map well into tissue microstructure. Again, we should expect fat to be smooth, homogeneous, low contrast, muscle to have strong directional dependence, mid contrast, and then trabecular bone high contrast, high dissimilarity, complex structure. And so then what we're trying to do is build a procedural histology engine, which is where we take these tissues and we look at it like a histologist would. So it's, kind of the sort of the standard for looking at these tissues and identifying them as tissues. So now what I want to do is take this and replace the GLC with a spatial pooler. This is where we have this spatial pooler, which actually is something they use in deep learning as an encoder, but also they use it in the hierarchical temporal modeling world. And so they use it oftentimes to pool data into these columns in hierarchical temporal modeling. So it was actually taking inspiration from the way that spatial poolers are implemented in hierarchical temporal modeling, because that's kind of a library that's easily interpretable for this purpose. So that's kind of what we're doing here. So we're importing things, importing this spatial pooler class. Then we are importing Perlin Noise plus this FBM generator we saw up above. Then we're running this, and we have this FBM Perlin 2D function. And then we're returning that. Then we have our spatial pooler feature extraction, which is where we instantiate the spatial pooler once or per image, depending on learning needs. For our genetic algorithm, we might want a fresh spatial pooler or a trained one. So we're going to get a fresh one per image for simplicity. So we have our spatial pooler features here. Then we have our new spatial pooler instance for each extraction. This is where this is here. Then we go down to tissue profiles, and their genetic algorithm, again, is defined. And we run our GA. So we have this 256 by 256 matrix running it for 40 generations, a pop size of 30, mutation rate of 0.3. And then we have the visualization. This is the visualization here. And now we're getting something a little bit better. We're getting the fat texture, which is, I mean, I don't know if that looks like fat. I mean, it kind of looks like it just has these bars that are going across. Now, notice we've kind of deviated from the Perlin Noise. It's generated in that same way, but it's not. It doesn't look like the original Perlin Noise too much. It looks like these bars. So the muscle is kind of striated. The fat is, I don't know. And then the trabecular bone is, I'm not sure that this is going to take the place of a histologist anytime soon, but this is what we get. This is the additional part of this. And so now we get these visualizations, and we see that the GA actually converges a lot sooner for fat and muscle, and then trabecular bone, it's around the same. So I'm not sure what to say about that. And we have our spatial pooler here. And we have so we have all that here. Not sure what that's running again, but what happens here is we have the output of our spatial pooler. So now this is, so spatial poolers take a bunch of data from different parts of an image, and it builds these sparse representations of the image. So it's taking things from the Perlin Noise or from these images, different regions, and it's building the sparse distributed representation of those spatial distributions. That's what a spatial pooler is doing. So it's doing this across the image. And so now we have the benefit of the spatial pooler. This is what we have here. So this is just showing what the spatial pooler is doing. So this is the original Perlin Noise input. Remember, we had this sort of wispy looking patterns, very blurry, but you can sharpen them up. Anyways, in this case, this is Perlin Noise is the input. It's taking this binarized input to the spatial pooler, so it's like segmenting the image almost. And then it's producing this SDR output into 45 active columns. And so then it's giving us this permanence map for column 0. It's reshaping this, and it's giving us this map. It almost looks like Perlin Noise, sort of a form of Perlin Noise where there are different permanence values across the matrix. So that's what this is giving us. Not sure what good this is going to do, but I wanted to see what it would do. And so this is, I mean, it might be useful, but again, it's like, you do these explorations and then you figure out what it's for and you kind of put things together. Okay, so then we have this 3D Perlin Noise generator. And what we're doing here, I believe, is our GANs. So this is where we're building a GAN, a three-dimensional GAN. Now I haven't finished this. This is not giving me an output right now. This is where we have our generator and our discriminator, where we're taking our training set, which are the Perlin Noise images, and we're just training our model on it. And then we're kind of presenting sort of adversarial examples, and we're training our model on it. So the generator is generating adversarial examples as well as regular Perlin noise, and the discriminator is trying to find the correct Perlin noise. Basically, that's what's happening. And then we have, we go through here, we have generator data set on the 3D volumes from the trained GAN. So it's generating 3D volumes. And then it's running that. And it ran 10 epochs, which is the thing you do in GANs at these parameter values. And then it says it saved things to GAN 3D volumes, but it didn't actually work. So I don't know why it didn't do that. It didn't There's no output directory, or there's nothing in the output directory, so I don't know what's going on. I'll have to play with it some more. It takes a while to run, actually, in a notebook. And then this final part is the generated 3D volume, where you have for the axial, coronal, and sagittal slices. So it's like the XY plane, the XZ plane, and the YZ plane. So it's just different views of this volume. And then we have this visualized slice grid, which visualizes this. And then the visualization parameters here. And then we have, and so then this would be the volume if it worked, where it would show you everything, but I can't get it to run. So I mean that's kind of what I've been up to with that. Um, I think we're pretty far from like a paper on it, but I think it's no. I wanted to follow up on it because it's. I think Dick was interested in this right before he died. He was like, "This is really kind of an interesting approach." So I mean, we did this chapter following up on the biomedical imaging aspect of it, but I think there's more to say. You could simulate tissues doing this using this kind of methodology. It's just not clear to me. Well, from right now, it's not clear to me how this would kind of translate into simulating actual tissues, but it simulates what they might look like or what their sort of statistical structure.
Not questions regarding this, but I had a general question. Like the midterm evaluation is coming up, so do I have to know anything about that?
Uh, I think you might. I do your evaluation first, and I think you have to respond or something like that. I'm not
Uh,
really sure.
I mean, this notebook I just I was going through it, but like it's mostly like bone tissue related, so I've not been in that field. So, but I understood some of those coding, like lens space and all. I've used those, so a
Okay.
bit of it I was understanding. Yes, that's it.
Yeah. Yeah. I mean, it's like basically. I mean, all it's doing is kind of modeling what it might look like, and it's like
Okay.
trying to get the parameters right. But, um, yeah, and it's gonna be weird. It's kind of like this thing that this old comment about like how in 10 years deep learning will replace radiologists, and it's been
Yes, yes,
longer
yes.
than that
Actually,
and they haven't been replaced. And this is kind of why you can see that like it's hard to get the it's hard to figure out what you know I mean, you can characterize like tissues and other things like that as statistical structures, but there's this aspect of the sort of the gestalt structure, the thing that like a human would see and say, oh yeah, that's what that is, that's a tumor, or that's normal tissue. That isn't really something that uh, AI can replicate, or computer languages can replicate. Yeah. Okay. Do you have any other questions or? Okay. Why don't we get on with the papers then? So this is something that I presented last time. This paper, could living cells use phase transitions to process information? And so what they were talking about was they were talking about how living cells have these structures called condensates, which are just bundles of biomolecules or other things like that. And they, people don't necessarily know what they do. Their range of functions there, um, and they're involved in a lot of different things, including phase transitions. So, like transitioning from like a stem cell to a differentiated cell, or going through like cell cycle or something like that. And so the idea being that these condensates are involved in these phase transitions, but in doing so, they process information. And so, in processing information, this is something that can allow a cell to solve problems because it processes information, it kind of knows what's going on in the cell, and then it triggers some process. And so, the idea is that it's processing information, much like maybe like a cognitive system. And then they call this embodied computation because it's dependent on being in the cell, being a condensate, or things like that. So that's what they talk about in this paper. I wanted to go through some of the figures because, you know, um, just to do it justice, if nothing else. So this is their model of living cells computing. So they're focusing on biomolecular condensates, and so condensates are these things here in green. So you have different concentrations of condensates in a cell. And so cells are always integrating different chemical signals. And so the chemical signals have different strengths. It's much like sensory information in humans. If you have visual information or auditory information or haptic information, it always happens to be different strengths. And then, we've built neural networks on this principle that we're dealing with things of different strengths, are, connections between neurons are different strengths. And then that allows you to process information. And so the idea is the cell here is taking in a bunch of different stimuli, multidimensional information, nutrients, growth factors, etc. And then it's providing a response, which is like differentiation, division, metabolism. These stress granules, these green spots here are stress granules. And so they are these fluid like biomolecular condensates. So you can see that they have an inner body and an outer shell, and they exist embedded within this tissue at different points, maybe along a boundary of different tissue types. So you can see that the condensates are sitting there and they're taking in, they're part of this information processing system. And they're almost acting to like as sort of way stations for the information. They can, transform that information into action, basically. People also argue that microtubules do this in different ways. So, the site of action is they're arguing here for condensates. Other people, argue for microtubules or even like actin molecules. But the idea is that those structures serve to sort of act upon that information. And so then this is thinking about biomolecular condensates and sort of the statistical mechanical framework. So in our Saturday meeting, Morgan and I were talking about the unreasonable effectiveness of statistical mechanics. And this is something that it's a catchy little phrase, but the idea is that you can model a lot of things with statistical mechanics. And, you can model all sorts of different phenomena, making it sort of this universal framework for looking at different things. So this is where we have this canonical system, which is where we have a system concentration and we have these different condensates, and there's a sort of a heterogeneity, there's a relative heterogeneity across this canonical system. So any one cell, we have a bunch of condensates, and we can take an ensemble of those condensates, and they're maybe in different states, or maybe they're all in the same state. And the idea is what we want to do is we want to count up those condensates and we want to survey their state. And then we want to return this sort of statement about how diverse or how homogeneous the system is. And the idea is if it's homogeneous, then it's sort of in a low energy state. And if it's diverse, it's in a high energy state. So this is where we have this low concentration and a high concentration where the condensates are in different states. And so we have our system concentration kind of acts as the energy. And then we have the grand canonical system. So this is where we have different phases where, you have the condensates actually doing something at a site of action. So this is again where we have this low concentration state or this low energy state, and then this high concentration state or this high energy state. And we can see that these condensates in this case are adhering to this surface. They're sort of forming the subcompartment. So, if there's a low concentration state, you have some of the condensates go down to this substrate, and they form sort of, some of them are sitting on the boundary, some of them are off floating around. There isn't a lot of sort of organization here. In a high concentration environment, you have this basically this entire substrate is filled with condensate particles, and you have the subcompartment that forms from that. And of course, when you have this kind of structure that forms from that, you get this information processing. It's sort of like hitting a threshold. So you see from phase A to phase B, you're shifting across the boundary and you're creating a phase transition there. So this is, again, thinking about information processing. So they make the analogy between what a neural network does in terms of classifying images, and then this molecular information processing where there are these phase transitions. So the neural network, of course, has synaptic weights, Wij. These are have different weights, and they determine the different sort of feature classes that are generated by different images of pets, like dogs versus cats versus something else. And you can see that there are these decision boundaries that get made in a machine learning context where a decision or a boundary is drawn, and it sort of, it defines these different groups because there's separability between the groups, because we have enough information that the neural network can act upon to draw this boundary in that separable area. Now, the same kind of holds true for the molecular soup, as they call it. And the molecular soup is also a network, but instead of using synaptic weights, it uses binding affinities. And it's the same weight matrix, Wij. And then you look at this and you see that there are these phase boundaries instead of decision boundaries, where you have these different ensembles, or these canonical ensembles in different parts of the space, and they're in different phases. So there's this phase boundary that forms from kind of where they're distributed in equilibrium, and then as they move around, they kind of cross these phase boundaries, triggering a phase transition. This is, of course, working with logic gates. So they're showing, separation of concerns-based computation. An example of this would be a number of logic gates configured in a network. So you have an OR gate and a NOT gate combining into an AND gate. And the idea is that you can have these different collectives of condensates, and they could form these metamaterials, or different cells can form metamaterials. So based on the state of these condensates. So you can actually program these cells in terms of doing things with the condensate distributions. But a way to build these metamaterials, or these collective modes. And so this is where I guess someone is saying 3 or 4, and the material is responding by changing its phase, and they're able to actually take the transmitted energy from their voice and just kind of shift things from 3 to 4. I guess that's the idea here. I don't think they've actually built anything. They're just showing a concept here. This is kind of showing some of the aspects of sort of the computational capabilities. So you have different concepts like sharpness of the gradient, capacity of the phase space, and then this expressivity, where how much can a computational system like this condensate model, how much can this encode and how much can it express in terms of different things that it allows the cell to do. So we talked about phase space and triggering these phase transitions. But what else can it do? Because we want to have a high degree of expressivity, and so we can do other things besides just kind of pushing the cell into a different state. And so there's this low expressivity environment where you have a linear boundary between phases, and high expressivity where you have this curved boundary between phases. And it's still doing a proper classification here. This is kind of like a classification boundary in a discriminative model. So this is like a linear regression, and this is a polynomial regression, basically, or a nonlinear regression. I mean, that's the analogy. What they're saying is that there's this phase boundary, and the phase boundary is linear here and nonlinear here. When you have a nonlinear phase boundary, then you have this high degree of expressivity. Okay, I think that's it for the figures. So that's that paper. I just thought it was really interesting to go through and see what they were going to talk about in that paper, and how they were going to show their ideas. And again, they've not implemented anything. This is just an idea of how a cell might do information processing. There's a whole literature on cellular information processing. Like I said, using different mechanisms. But the idea is that it's taking these environmental signals, it's transducing them to some sort of chemical concentration in the cell, and then there's some mechanism that's acting upon that to do different things. And the idea would be you can control this maybe to program the cell to do specific things. So people have wanted to do all sorts of things related to biomedical engineering or other types of applications where they can control and steer the cell to different locations or control the fate of the cell or things like that. So this is another paper that kind of gets into a similar area. This is called Decoding the Architecture of Living Systems. And this is just kind of, this is actually a long paper that goes into, things like complex networks and dynamical systems theory and evolutionary dynamics. And it talks about how how can we model living systems? How can we understand them? How can we understand not just their component parts, but their behaviors? And so that's what this paper is about. And so it's a really long paper. I'm not going to get into it, but I do want to. This is the outline you can see. But I just want to go through the abstract and get you thinking about this area. So the abstract reads, the possibility that evolutionary forces, together with a few fundamental factors such as thermodynamic constraints, specific computational features enabling information processing, and ecological processes, might constrain the logic of living systems. This possibility is tantalizing. However, it is often overlooked that any practical implementation of such a logic requires a complementary circuitry that, in biological systems, happens through complex networks of genetic regulation, metabolic reactions, cellular signaling, communication, social and eusocial non-trivial organization. So a lot of things from like these abstract networks to different types of communication and types of organization. So it's a broad swath of things. And this is one of the things when people will often talk about how biology is complex. but that doesn't really describe anything. So we want to really break that down and get to the fundamental components. And it is hard because there's so many sources of variation here, so many sources of complexity. It's hard to find the thing that will allow you to simplify that. Maybe for controlling the cell or predicting what it'll do or, being able to replicate it in some way. Here, we review and discuss how circuitries are not merely passive structures, but active agents of change that, by means of hierarchical modular organization, are able to enhance and catalyze the evolution of evolvability. So, evolvability is a term, or it's an area of biological theory, where the argument is that we want to be able to have a system that can evolve at a certain rate. So evolvability just means that something can evolve, something has a space of possibilities to which it can evolve. Now, some things aren't very evolvable. Some things in this area of theory, people look at different types of systems and they'll look at how evolvable they are. If you don't have the ability to be plastic, some organisms are not very plastic, others are highly plastic. That means that your evolvability is limited. Or if you are have an evolutionary history that constrains you to a highly specialized ecological fate, you may not be a very evolvable species. That's just there are certain things that are more evolvable than others in the history of life, and people want to know why that is. And so there are all sorts of things having to do with standing variation and the response to change of organisms that go into this idea of evolvability. And then it can also evolve itself. So sometimes evolvability is controlled by different genes. So if we think of like Hough genes control developmental pattern formation. But if you don't have those Hough genes, or if you have too many highly specified Hough genes, you can't evolve, say, like a new body type. If that's a key to survival, then you're toast. So, but the thing is you could evolve a new body type. I mean, it's harder to evolve than just kind of modifying the existing body type, but those things can evolve as well. So sometimes organisms or lineages are less evolvable, and their genes controlling their, say, degree of plasticity, and then those genes or those genetic networks diversify, they mutate, they change, and then that allows for more evolvability. So this is kind of this idea of evolution of evolvability that is key, I think, to understanding some of this complexity. Because people will just say, well, these are the things that, a laundry list of things that are complex about a cell. But if we think about it in this way, we think about it, well, okay, what of those things make the cell or the species evolvable? And then how can that be controlled by different genes? And then we can understand maybe the complexity in a little bit better way. By analyzing the role of non-trivial topologies in major evolutionary transitions under the lens of statistical physics, again, we're using statistical physics, and nonlinear dynamics, which is, the theory of computing trajectories for things and looking to see if there are attractor basins for things, meaning if things have a tendency over the course of those trajectories, and then analyzing that in the context of continual measurement. What is that's one way of doing this. Statistical physics, like I showed you with the ensemble technique, that's another way. Using these different types of lenses, looking at different major evolutionary transitions, we show that biological innovations are strictly related to circuitry and its deviation from trivial structures and thermodynamic equilibria. So, when we get an innovation, like say we get a new type of brain in evolution, this is, of course, related to circuitry, so we have to have the circuitry, the sort of the locus of evolutionary change or of plasticity has to be at the circuit level. And then we can build this brain, we can build the tissues, and we could build the behaviors from that. And then, the idea is that so from its deviation from trivial structures and thermodynamic equilibrium means that the system is out of equilibrium, and it's not building trivial structures, it's building the circuitry. So biological innovation is, we can tie this to specific changes in circuitry over time. We argue that sparse heterogeneous networks, such as hierarchical modular, which are ubiquitously observed in nature. So hierarchical modular is where you have a type of network that has sort of a root, and then it splits into two parts, and those two parts are modules, and then it splits into another set of parts, which are also modules. And there is this mother-daughter relationship between the nodes, or a mother-child relationship between the nodes, that's hierarchical. So one category is sort of the precursor for another, or one category controls another in the case of genetic regulatory networks, and so on and so forth. If we think about the C. elegans lineage tree, that's hierarchically modular. We start with a single cell. That divides into two cells, which define the poles of the embryo, and then everything underneath that is within those poles and their specializations. So hierarchical modular networks are favored in terms of their trade-off between energetic costs for redundancy, error correction, and maintenance. In other words, whatever it is that you're looking at in life has different energetic costs for different things, like redundancy, meaning how many parts do the same thing, error correction, meaning how many things got to correct for aberrations, and then maintenance, which is how many things are devoted to keeping things alive. And so there's this trade-off that we see there. There's an interesting area of evolutionary trade-offs that this fits into as well. We identify three main features. So three main features are interconnectivity, plasticity, and interdependency. So we do have plasticity in there. We also have interconnectivity, which is where things are connected together, and then interdependency, which is where one thing is dependent on another thing. So those three features point towards a unifying framework for modeling the phenomenology, discussing them in terms of dynamical systems theory, non-equilibrium thermodynamics, and evolutionary dynamics. So as you might imagine, evolutionary dynamics is also important here. And evolutionary dynamics really focuses on, how things evolve. So how do populations evolve things? What are the components that are needed for innovations or keeping things stable? Or, especially if you're interacting with another population, what does that result in? Within this unified picture, we also show that slow evolutionary dynamics is an emergent phenomenon governed by the replicator-mutator equations as a direct consequence of a constrained variational non-equilibrium process. Overall, this work highlights how dynamical systems theory and non-equilibrium thermodynamics provide powerful analytic techniques to study biological complexity. So there are a lot of big words here, a lot of topics that, sort of span evolutionary theory and statistical physics. So this is something that, again, you have to really dig into to fully understand. So I'm not going to go over this too much more, except to say that I think it's an interesting kind of stab at this theory. But this is, again, the types of things we have to think about when we think about, organism modeling or even tissue modeling. Okay, last thing I want to touch on here is this new paper that came out in Frontiers in Neural Circuits. This is probably interesting to everyone in this group. It's a review article. It's a review article, and it is called "From Small Brains to Smart Machines: Translating Caenorhabditis elegans neural circuits into artificial intelligence." So this is thinking about C. elegans connectome as an artificial intelligence system, and they're not going to implement anything here. They're just kind of, you know, going through the motions on this and trying to propose a way forward. Of course, this is the kind of thing OpenWorm is trying to do in some ways, and so they're just laying out a roadmap here, I guess. So let's get into the abstract here. So the hermaphroditic C. elegans, with its fully mapped connectome of 302 neurons, offers a paradigmatic example of how a minimal nervous system governs biotic, adaptive, and context-dependent behaviors. In contrast, modern artificial intelligence systems achieve intelligence through scale rather than efficiency. So this is like the implication is that C. elegans is all about efficiency, where we have a minimal nervous system governing all these diverse behaviors. But in artificial intelligence, we're just throwing scale at the problem rather than efficiency. And of course, as we know, scale is a problem because you need a lot of compute. And so, you can't just throw compute at something. Sometimes you need to really think about efficiency. So relying instead on massive data sets and artificially engineered architectures. So again, this is a common theme that people want to get away from that, but they don't know how. We also have massive training sets for AI, whereas in C. elegans, we have a lot of innately coded things in the behavior. So we build these circuits, and the circuits sort of provide output. You have a system where if you take in environmental information, outcomes, behaviors. So it's a very generative model, but it doesn't rely on a lot of training. I mean, in humans, of course, in human babies, in human babies, you get limited exposure to things, you get limited experience. And from that, you can actually learn quite a bit. And then you learn more and more and more. But you're using, in terms of your training data, you don't have to have, as much training data as you would for an artificial system. So you don't have to learn everything about a topic to learn something about the topic. You can extract things about it. You can sort of get the gist of things. And there's this same kind of holds true for C. elegans, although C. elegans is more sort of deterministic in its behaviors. But in any case, you don't need that kind of training data. Okay, so this mini review explores how C. elegans neural circuits can inform the development of more efficient and flexible artificial neural networks. We highlight recent studies that translate the principles inherent to C. elegans neural circuits into artificial neural network architectures, with applications in machine control and image classification, resulting in enhanced robustness and improved performance. By distilling neural principles from the simplest known nervous system, this mini review outlines a pathway towards compact, adaptive, and biologically inspired artificial intelligence systems. Okay, so this just kind of goes through a review here where they talk about ANNs and how they're all the shortcomings compared to biological connectomes. And so then it just talks about the C. elegans nervous system and how this works. And it's, just kind of talking about the relative simplicity, although I don't know if it's relatively simplistic because you have, let's see, so it says, although the nervous system of C. elegans already has a minimal structure compared to most known biological neural networks, meaning connectomes, it can further be compressed via systematic abstraction and consolidation. This approach is based on the observation that the neural network contains functionally redundant neuron clusters. So we build this functional equivalence framework where each homogeneous neural cluster is collapsed into a single functional node. So they're building a network where they're taking these homogeneous neural clusters and they're collapsing them into a node. It's almost like a hypergraph, but they're using just a sparse representation instead. And so all synaptic edges originally connected to the neurons within the cluster are then merged into the corresponding node under a weight-preserving constraint. Ultimately, structural consolidation streamlines the neural network to only 122 functional nodes and approximately 2,400 synaptic edges. So this is actually still quite a bit of connectivity, but we're collapsing our connectome from 302 nodes to 122 nodes. So this in turn generates a compressed neural network that preserves the key topological and dynamical properties of the original network. So then they say, okay, then they build this sparse representation of the connectome. Then they say that there are these partially segregated layers that one with sensory interneuron motor hierarchy. So this is, again, what we talked about last time. We have these sensory neurons, motor neurons, and interneurons. So we have these layers in the C. elegans nervous system. We have the sensory neuron layer, the interneuron layer, and the motor neuron layer. Motor neurons drive behavior. Sensory neurons take in inputs from the environment. The interneurons connect them. So that means that we have further structure here that we can use to guide behavior. This allows flexible computation through distributed processing rather than centralized control. So for the directed and weighted network obtained through the reconstruction of C. elegans connectome, we can get a detailed analysis of topical organization, weight heterogeneity, and hierarchical modular structure. And these are the core indicators that characterize the structure of directed and weighted networks in network science. Node strength is used to measure the total strength of a node's outgoing and incoming connections. In strength and out strength quantify a node's ability to be controlled and to drive other nodes respectively. So you have this aspect of controllability. So if you're familiar with network controllability theory, this is important because oftentimes in a connectome, as maybe in also an AI system, you would want to have a sort of directed behavior where something is controlling something else. It might be controlling a muscle or a robot, and we'd like to know where those nodes are and how they function. So this is thinking about network controllability. This is a graph that shows, I guess this is the connectome here of C. elegans. And it just talks about these different connections that you see in the connectome. I don't think it provides an image of the reduced network, but just imagine that all of these nodes are reduced down to a smaller set. And then B, of course, are these layers. So you have the sensory neurons, the interneurons, and the motor neurons in this three-layered configuration, with pharynx neurons being like sensory neurons going into the interneurons. So the idea here is you have these layers like you have in a neural network. Okay, so they analyze the connection strength, in strength, and out strength, and they show that only a small number of neurons in C. elegans neural network possess high connection strength, suggesting they may be core neurons regulating life activities. This is consistent with the concept of scale-free networks in network science. Thinking about the analysis of edge weights, this shows that only a small number of connections have large weights, further reflecting the heterogeneity of synaptic connections. The shortest path length of analysis in figure 2e indicates that the shortest connection distance between most neurons in the C. elegans neural network is short, demonstrating high overall information transmission efficiency of the network. So this means that we don't the transmission distance of the connections are short, so you don't lose a lot of information over the course of transmission. Analysis of betweenness centrality in figure 2f shows that only a small number of neurons in this network exhibit high betweenness centrality. So it means that there isn't a lot of small world structure to the network. It's just scale free. And then this feature is consistent with the analysis results of neuron connection strength, further confirming these neurons play a core role in the network and support the stable operation of life activities. And so they kind of get into this analysis. They talk about small world networks. And of course, we have small worldness in the C. elegans connectome, but not a lot. It doesn't dominate. Its core architecture exhibits the coexistence characteristics of dense local connections and a small number of long-range connections. This unique configuration can simultaneously improve information transmission efficiency and enhance network robustness, providing a structural basis for the nervous system to stably perform physiological functions. So that's a little bit about the network. Now thinking about the nervous system being a complex network composed of a series of nested neural circuits. And that means that there's this information processing logic that we actually saw in the last paper where they were doing these tests on different types of circuitries and different types of logic gates. And then this is a nice figure that shows the metrics of the nervous system in terms of edge weights and node strengths, and then this rank order analysis, where you see this sort of power law distribution, where you have some cells that have a high node in strength, a high node out strength, and a high overall strength. And you see that it's the same sort of, you have ALA, AVA, VD, CAN, PVD, RIA, RMD. So these are all cells that are commonly, have a high node strength, both in strength and out strength. And then you have the edge weights. And you see that the same neurons are kind of involved in this as well, or were, most of the same ones, where you have high edge weights and high betweenness centrality. And then there's this length of shortest path graph in E, which is this power law. It's kind of not, well, I guess it's power law. Yeah, it's a regular power law where you have this set of paths and then you have longer paths. So you have very few paths that are long and a lot of paths that are short. So that means that you have this sort of maybe small worldedness in the connection length, but scale freeness in other ways. So the point is that it's not structured like the type of neural network that we would use for machine learning. And so that's kind of why they do these analyses to show this. So they do some behavioral experiments, or they at least simulate some behavioral experiments where they're actually looking at how C. elegans learns and how you can change the network. You can ablate different cells to change the behavior. And again, this is, you could do these kinds of experiments in neural networks, but the credit assignment isn't quite the same. And I would argue that if you go outside of C. elegans to other connectomes, you don't get this sort of ability to do this kind of these ablation studies, because single cells in C. elegans, as well as some insect connectomes, are highly specific to their function. Whereas if you go to mammalian nervous systems, you can ablate cells and other cells will pick up the slack. But if you ablate like a region of the brain or some structure, then you have the same effect. Where if you take out that part of that network, then you lose function. And sometimes it's very selective, and sometimes it can be overcome, and the network is robust to that ablation. So that's kind of what they're getting at with here, making the point that you can, that the architecture matters, that sometimes there's a robustness with respect to the architecture, and sometimes it's quite brittle, and it's very different from maybe what you would see in a neural network. So this is where they translate neural circuits to AI design. And so there's a study by Lechner where they take the nervous system of C. elegans as the core biological prototype, and they propose an engineering strategy based on its neural circuit. So this is Lechner, Matthias et al. 2020. The strategy effectively addresses the key bottlenecks of traditional ANNs and autonomous vehicle control scenarios, namely parameter redundancy, poor interpretability, and insufficient robustness. So the core innovation lies in the accurate replication of the hierarchical topology and sparse connectivity characteristics of the C. elegans connectome. The C. elegans nervous system achieves complex behaviors such as motor control and environmental navigation through its hierarchical topology. So, this hierarchical nature of the connectome is key here. The sparsity of its neural synaptic connections reaches 90%. Even though there are a lot of synaptic connections, it's still a sparse representation of full connectivity, an example of full connectivity. So the sparsity of its neural synaptic connections reaches 90%, and there exists strong recurrent synaptic connections between interneurons and command neurons, enabling it to balance information integration efficiency and decision-making response speed simultaneously. And so the C. elegans nervous system possesses core computational advantages such as distributed control, temporal dynamic adjustment, and maximum information transmission efficiency, which can be migrated directly to the architectural design of autonomous control systems. So they were using this for autonomous control systems, and they used this in lieu of a neural network, and they were able to get good results. And the reasons for that involved in the sort of topology of the network. Now, in terms of design, the study converted topological dynamic characteristics of C. elegans neural circuits into a trainable ANN architecture, which specifically includes two core designs. So the design features are neuron model innovation, where they used a liquid time constant neural model. Then two is network architecture reconstruction. So the front end uses convolutional layers to extract high-dimensional environmental input features from autonomous driving scenarios, compressing them into 32 feature dimensions, corresponding to the sensory neuron input of C. elegans. So they have these two different things. They have this type of neuron model, liquid time constant model, and then they have this network architecture reconstruction, where they have these convolutional layers that are used to extract high-dimensional environmental input features. So this is, then they have this other study, Wang et al., where they expand the engineering application scenarios, extending its application field from the previous autonomous vehicle control to the computer image classification task. And in this case, we have two factors here, where we have this topological function matching of neural circuits. So this pathway from sensory neurons to interneurons to motor neurons is functionally mapped to the core process of image classification tasks such as feature extraction, feature integration, category output. The second part of this study, as opposed to the last study, is by using reorganization and optimization of network modules. So this is where you basically have these convolutional layers, pooling layers, fully connected layers, all used in image classification in neural networks. In the case of C. elegans, the C. elegans analog, we have module reorganization and connection optimization performed based on the topological structure and information flow direction of the C. elegans neural circuit, ensuring the neural network architecture aligned with the efficient processing logic of biological neurons. Then they have the elegans AI model by Bardozo. This is where you're using C. elegans as a way to enhance the performance and efficiency of artificial neural networks. They design two core model architectures. There's M1, which is based on transformers, and then M2, which is an autoencoder inspired architecture utilized for the unsupervised image reconstruction task on the MNIST dataset. So the first M1, this transformer-inspired framework, used CFAR 10 as the benchmark, and M2 used MNIST as the benchmark. And so they did these comparative experiments where M1 achieved a top one accuracy of 99.99% on both CFAR benchmarks, and M2 attained an accuracy of 99.84%, in the unsupervised reconstruction task of the MNIST dataset. And so they saw some success here. Furthermore, the model based on the original C. elegans connectome outperformed those based on randomly rewired and simulated connectomes. So the topology definitely has an effect, and you can use random rewiring and other techniques to sort of get a sense of what the random state would look like, and then use C. elegans as sort of the experimental method as to what the structure might look like. And so they were actually able to get good results out of the C. elegans example and not so good results out of the randomly rewired example. So it's the structure of the C. elegans connectome that's really giving us an effect here. Okay, so that they have a number of different other studies that were done on this. So people have actually looked into this quite a bit. I think this is a really good paper for kind of surveying this area to see how people are kind of experimenting with C. elegans connectome and throwing it at conventional machine learning problems and doing pretty decently on them. So I think I'm going to leave that here. And that's all for today. Okay, so thank you for attending, and see you next week.